be getting started in the interest of time. First of all, I'd like to acknowledge the traditional owners of the meeting today and to pay my respect to the elders past and present. My name is Natalia Gorobudi, and I'm the new director of the Cog Neuroscience Hub, based in Malta. We are going to have a few of these research seminars throughout the year, but this is our first meeting for 2026, and I'm very happy at how many of you come. And that's probably because we have a really excellent speaker today. We have Thomas Andrillon, who's a neuroscientist at the INSERM, which is the equivalent of the French NHMRC, and he is also at the Paris Brain Institute in the DREAM team. I love the name. So the Sleep, Dream, and Cognition team. So Thomas obtained his PhD in 2016 at the Neurodegenerative in Paris, and then moved to Sydney, to the University of New South Wales, to do a postdoc with Professor Joel Pearson, and then to Monash to do a postdoc with Professor Nawat Suchia. And you have also done some lecturing at Monash, right? But he'll tell you more about his trajectory a bit later. And we'll let everyone come through. Yep, so join places. And without further ado, to you, Thomas.
Great, thanks. I'm visiting Australia after a 5-year gap. So it's really great to and it's really great to have the opportunity to present some of what I've been doing. So today I will focus on mostly on wakefulness, but I'm initially by training a sleep scientist, a neuroscientist. But you will see that I approach wakefulness with this sleep perspective. Before starting, I wanted to do some sort of a self-introduction because the topic of the talk is related to actually my trajectory. So as Natalia said, I was trained in mainly 2 places in Paris, where I was not born, but educated, and at the University of Wisconsin, because I joined the laboratory of Gio Tononi and Cara Cirelli, who explore consciousness through sleep, and that's how I got into sleep research. I spent then quite a long postdoc / research fellowship at Monash University, with a bit of a time at UNSW. It was a bit complicated. I enjoyed a lot the community in Melbourne. I couldn't put all the people I started collaborating with, but that's one of the things I think that's for which I wouldn't say you're lucky, because you built it, but this sense of community and the fact that people are collaborating is really unique compared to other places. So it was really great for me. Before arriving to, now I'm in the Paris Brain Institute, which is kind of Flore Institute, so it's a big neuroscience institute. There is some cognition in it. We're not the main components, but when you look at the teams in cognition, they're quite strong in terms of impact. And we just created a new one, which is called the Dream Team. So I didn't pick the name, but it's memorable. And we're actually 4 people in that team, 2 medical doctors, Isabelle Arnouf and Pauline Debray. And we're 2 basic researchers, so with Delphine Diet. And we're very interested in sleep, dreaming, and the link with cognition. So before arriving in Australia, it was after our PhD with my wife. We wanted to do a bit of a break. So we went for quite a long honeymoon trip. So we took a car, basically drove down to Patagonia and back. And you can imagine it's a pretty long drive, even for Australian standards. And I did something that I don't advise, which is I put the car on the roof somewhere around here. So accident over an accident. What happens? Okay. I think it's okay. So yeah, stupid accidents, just material damages, luckily, nothing too serious for us. But after that accident, obviously locals were a bit perplexed with the thing, and they asked me what happened. Did I fall asleep? Did I drink? Did I hit a llama, which is one of the three main sources of accidents? And it was neither of the things. The only rational explanation was just thinking about something else, which was very, very difficult to explain. So we ended up going for the llama explanation. People are very sympathetic with llama accidents. They go, oh yeah, that can happen. So yeah, I'm wondering if it's just I might have too many things open. So after that accident, it's really where I started getting interested in mind wandering. I mean, why in some situation where we're really not supposed to think about something else, we still think about something else. Why does it happen? How does it happen? Etc. etc. So that's a lot of the research agenda I followed when I was in Monash and now that I'm in Paris. And I mean, I'm obviously not the first one to ask this kind of question. Back to William James, there's this wonderful metaphor of the mind being like a bird alternating between flight and perchings. So we can focus on something and then we'll move to another idea and another and another. And these thoughts or experiences we have are not necessarily related to the here and now. That's the magic of our brain. We can think about the there and then, even if sometimes it means not being really connected to our environment. So classically in the literature on mind wandering, there will be this division between being on task, so focusing on what you're supposed to do, versus mind wandering, thinking about all sorts of things. Recently came a third option, which is the notion of mind blanking. So the fact that the stream of consciousness is not necessarily continuous, you don't necessarily jump from content to content. Sometimes you can have a moment where, well, you're still there, you're still conscious, but you are not able to report anything in your mind, which is what I will refer to mind blanking. Usually, when I talk about mind blanking, there are two reactions in the room. There are people who get exactly what I mean, and then people who are wondering what this is. So I think that a lot of people will experience mind blanking in their everyday life. And for some others, it's something maybe a bit weird. And to be honest, we're still working on the description, conceptualization of that phenomenon. If you are interested, after a symposium at the annual meeting for the Association of the Scientific Study of Consciousness, the ASSC, we wrote a review on the phenomenon of mind blanking, where we try to actually, well, get into the weeds and how to define what it is. But it's a very, I mean, the first paper on mind blanking was not in 1984, it was in 2000, maybe 13 or something like this. So a very, very recent investigation. So you have this notion of the stream of consciousness where your experience will alternate between being on task, being mind wandering, maybe being mind blanking. And this is usually separated from the study of sleep because we have this tendency to study separately modulations of the contents of consciousness and modulations of the states of consciousness, which is really things that's two orthogonal things. What I'm trying to do with my research is actually to see that as more of a continuum. Depending on your level of arousal, you can have small fluctuation that will make you think about something else, being focused on the task. But if the arousal decrease, you will start being more and more sleepy, and you might fall asleep, and you'll have other types of modulation of your conscious contents in that period. You might be dreaming, you might be focusing on your environment, because you can still be connected to the environment while you sleep, or you might be not conscious at all. So I'm really trying to put all of these different states on a continuum and trying to understand if there are similar mechanisms that can explain variations or modulation or transition between these states. Again, I'm not the only one going in that direction. There has been proposal, like the one of Kalina Christoph, of trying to put mind wandering and dreaming on the same, let's say, continuum with different models. of neurophysiological constraint that will determine what you're doing at any point in time. This view is supported by the fact that when you look at the subjective experience associated to mind wandering and dreaming, often it's pretty similar. It can be actually difficult to for a blind scorer to know if the report we're seeing is a mind wandering report or a dreaming report. And there are also overlapping neuroproblemations. This approach is also aided by a parallel change in the way we conceptualize sleep. For a very long time, sleep was divided into very rigid states. You have wakefulness, you have non-REM sleep, which was usually associated with dreamless sleep, and REM sleep, which is usually associated with dreaming. And there was this very convenient, some sort of bijective relationship. When you're awake, you're conscious and conscious of your environment. When you're non-REM, you're unconscious. When you're REM, you're conscious, but of something that is not the environment. This is evolving quite a lot. And if the slides go back, yes. I'm not sure.
Oh, my slide is too annoying.
No, I think it might be some. I want to just.
Maybe it's your computer mind blanking.
Yeah. Might want. Everything's well older than annoying parts. Yeah, so you have this concept of local sleep. Local sleep just means that transition between sleep states are not necessarily all or nothing. You can have transition states, mixtures where you have a combination in time and space of different type of brain activities. So, a classical case is somnambulism or sleepwalking. It's just a state where part of your brain is awake, part of your brain is asleep. And that's how you can have both features of wake and sleep at the same time. We see that in sleep, and I will show it that we see it also in wakefulness. So we are not in this situation where we're either asleep or awake. We can be in a situation where you can have what I would call intrusion of sleep-like activity, usually slow waves, in wakefulness. Why I'm interested in slow waves is because slow wave first is a hallmark of non-REM sleep. So when you fall asleep, your brain changes neural dynamics toward more synchronization and the expression of large amplitude slow waves. These slow waves are the largest physiological rhythm your brain is able to produce. And these slow waves are usually just a reflection of an interruption in neuronal activity, as if you have a bit of a gap, a moment of silence across vast assemblies of neurons. So you can imagine that when this kind of phenomenon occurs, it's very difficult to maintain a continuous processing of information or perhaps a continuous stream of thoughts. These slow waves in wakefulness have been observed in the context of sleep deprivation, for example. It seems like we have a real relationship. They have been associated, yes, with sleep deprivation. So if you push people to their limits, you will start seeing these slow waves, and they are not without consequences. They are associated with, for example, behavioral errors in a task. What I will present first is the work I did actually at Monash on whether these slow waves can also be observed without any sleep deprivation. So can we observe them in a normal person going about their normal day? So perhaps you right now. And whether these slow waves are not only associated with behavioral consequences, but also modulation of subjective experience, like mind wandering. So this is something we did with Nao Tsuchiya and Jenny Mintz. What we did, we tried not to get into too complicated stuff. We picked the most boring task on earth, which is a sustained attention to response task. So the classical version is just a series of digits occurring randomly on the screen, and you have to press a button each time you see a digit, except if the digit is a digit 3. So it's a go, no go task. There's lots of goes and a few no goes. So the first paper on the start was titled Oops. So it's a oops task because you will frequently press on the no go because you just get carried away. We had a phase version because, I mean, it's difficult to do a very simple paradigm with now. It has to be complicated at some point. So during that task, we had what we call the experience sampling. So every minute or so, we interrupted the task and we just asked to people what they had in mind. And thanks to Jenny Wins, we tried to get a bit into the granularity of things without making things too complicated. So they had a series of questions. Here I will focus on the first question we asked, which was the mental states. And they had to pick 4 options. I'm putting 3 because we merged 2 of these options. They either have to report whether they went on task, whether they were mind wandering, so something about thinking about something else. And we had quite a broad definition of mind wandering here. This something else could be something endogenous, like your shopping list, your next vacation, but it could be also the environment, like this very distracting view. So it could be anything that's really not related to the task. Okay. And then we had mind blanking. Mind blanking was actually 2 options, mind blanking or I don't remember, and we merge the 2 things. So they had these 2 options, but they rarely picked I don't remember. So we just merge it with mind blanking. Like, so if you look at what people report in this kind of task, well, pretty much half of the time they will be on task. But then they can be either mind wandering 40% of the time and mind blanking 10% of the time. You can see the dots are the the individual data points, sorry. So you can see there is a huge variability. I mean, it goes from people always on task to people that are almost never on task. So I think that there is a response bias. I mean, it depends what yourself will call I was mind wandering or I was on task. These states are always a bit of it's complicated. So we task our participant to give the best reflection of their mental state, but you can have differences. But as you will see later on, these differences are not just arbitrary, and we can see also inter-individual differences that map with clinical disorders. So, I mean, there's also maybe meaningful inter-individual differences here. These phenomena are not stable in time and they evolve. Usually people start the task pretty on task and then it degrades. You can see even that at the end of a block on the 10th probe, they know it's the 10th probe and you have some sort of a regain of attention. So there is motivation component and so on. Obviously, being mind wandering or mind blanking has an impact on the behavior. So here people had less correct response on the no-go trials. So they made more false alarms. What was particularly interesting is the analysis of the reaction time, because this time we could see a dissociation between mind wandering and mind blanking. Because in the case of mind blanking, we saw that participants had a tendency to slow down. While for mind wandering, they had the tendency to accelerate. As if mind wandering was associated with a state of impulsivity and mind blanking to a state of sluggishness. To us, this was very important because it showed that it's not just them giving random answers to this question. I mean, in their behavior, so quite implicitly, we see a reflection of differences between these mental states. Both mind wandering and mind blanking were associated with signs of hypovigilance and low arousal. And that was reflected either with objective measurements like pupil size, but also subjective measurements, vigilance scores. And it seemed that it's a graded thing where mind wandering, you're a bit sleepy, and mind blanking, you're even sleepier. So what we did was to look at the EEG, and we have a marker, slow wave, that we wanted to examine here. So what we did was to take the EEG signal, filter it to remove the fast frequencies, and detect these negative peaks in the signal. So you will detect lots of things, small peaks, big peaks. So what we did was to collect them all and examine the distribution of the amplitude of these peaks and put some sort of threshold, a very arbitrary 10% threshold. We did this because in sleep, these slow waves are defined with an absolute threshold that has been hand-voted at some point in sleep conference. So something that's clearly not very robust. So we had to come up with a definition of at which point a slow wave is of a high amplitude enough, so it constitutes something of interest. So we just focus on the top 10%. But this is completely arbitrary, and I hope that one day we'll be able to refine this definition, especially, for example, with the help of intracranial recordings, where we could look at actually neuron firing and we could confirm that this slow wave corresponds to a dip in neuronal firing. This has been done actually by a paper from Laurent Sheibani in Nature Communication. So we know that these waves in wakefulness correspond to a drop of neuronal firing. What we don't really know is kind of the amplitude drop relationship. So at which point the slow wave is big enough that it counts as something sleeping. So after this this procedure, we looked at these slow waves. I mean, they are not entirely like sleep slow waves. They share similar features, especially this large negative peak. If you are used to ERP studies, I mean, a 10 microvolts ERP is large. So they're quite large events. They start fast and they slow down. And I think we think it's related to the fact that these waves are actually a reaction to a bit of hyper excitation at the cortical level, and it's just shut down. So phenomenon, I will talk about that later, called cortical bistability. Very importantly, the slow wave tracks for measures of vigilance, both at the pupil size level and vigilance rating. The lower the pupil, the more you have the slow waves on the scalp. The lower the vigilance ratings, the more you have the slow waves on the scalp as well. So what we did then was to use these detected slow waves, which were detected at the central level, as some sort of predictor of behavior. So this slow wave lasts for pretty much a second. Trials last for pretty much a second. So for each trial and each electrode, we could have a label of presence absence, and we could use this binary variable to determine whether the presence of a slow wave was predictive of a certain type of behavior. So we can use different behavioral variable, whether people missed a trial, whether people did a false alarm. And what you could build is a statistical map of when these slow waves are informative about a specific type of behavior. So very much like what you will do in fMRI, in EEG. This is less common, but it's pretty much the same. So we could build these maps where we can have this relationship between the presence of a slow wave and behavior. So if you focus on false alarm, for example, you can see that slow waves occurring in the frontal electrodes are predictive of false alarm and positively predictive of false alarms. This is interesting because of the potential involvement, the potential localization of this slow wave over prefrontal cortices and the involvement of prefrontal cortices with executive function. When you look at misses, you have actually a very, very different pattern. This time it is the slow wave in the back of the brain, the over visual cortex, that are predictive of the misses. This was a visual task, so it tracks. Also the fact that it's lateralized to the right tracks to the fact that half of the stimuli were faces. So it seems that we have a good co-localization with how these slow waves impact behavior and the networks involved in the production of that behavior. Even when participants were correct, they didn't do a mistake. We could predict how fast they were with these slow waves. And when slow waves occur toward the central frontal areas, we can predict an acceleration of behavior. And when it occurs in the back of the brain, a deceleration of behavior. So it seems that we are, in very, very broad terms, that we have some sort of differential outcome with the presence of slow wave in the front or at the back of the brain. In the front of the brain, this wave seems to be more associated with markers of impulsivity, false alarm, shorter response, while in the back of the brain, it's more sluggishness. When you actually look at temporal relationship between these slow waves in front and the back, it seems that these slow waves start in the front and sometimes propagate toward the back. So when we see a slow wave in the back, it actually seems to be coming from the front, so kind of a more global slow wave event. These slow waves are also predictive of mind wandering and mind blanking. So in the front of the brain. What was interesting in the case of mind blanking, we have slow waves that seems to extend more toward the back. So we're back to this notion that mind wandering is getting a bit sleepy, mind blanking is getting a lot sleepier. So you have lower arousal and slow wave that becomes more and more global. So you're getting closer to sleep. So we have this model where we have one simple mechanism coming from sleep, slow waves, that could explain actually a lot of different behavioral consequences depending on where it is observed. So it's some sort of functional switch. It will just switch off temporarily a network, and the consequences will depend on whatever the network does. And if that network is not doing anything, then the slow waves are not are without any consequences. So how this leads to mind wandering and mind blanking? This is a very recent study, because it was published 2 days before Christmas, where we took the Melbourne data acquired here. We added some data from Paris, and we first of all replicated the behavioral results of the initial study. So that was a very nice self-replication in a way. So we have these markers of impulsivity associated with mind wandering, this marker of sluggishness associated with mind blanking. And we extracted then electrophysiological features that are used in usually disorders of consciousness to track levels of consciousness. Here we wanted to see if there could be also useful to track modulation of conscious contents within the wakeful states. So we have spectral features, complexity features, connectivity measures, and also we looked at more classical PRPs. This is a big, scary slide, but it's just to show you that we have a lot of interesting findings in this paper. If I have to sum it up, I would say that mind wandering here was associated with increased delta power, decreased complexity over frontal electrodes. We also have kind of a breakdown in frontal parietal connectivity. Mind blanking, we have also this increase in slower frequency, but in the back. So we still have this front-back contrast. The ERPs were particularly interesting. So these are maps of the responses to the digits. And what you can see is that in the case when they were on task, it starts with the back of the brain. And then you have this P300 phenomenon, very classical of conscious access and everything. When they are mind wandering, you still see the same starts. The end is a bit changed. It's not exactly the same pattern. You have a more frontal involvement. But in the case of mind blanking, you can see that the later part is almost cancelled. Actually, in the later part, all we have is this tiny dots on the left part of the brain, which is most likely associated with the fact that they were doing right-hand responses. As if in the case of mind blanking, the sensory information is directly rooted to the motor areas, and it's never really entering this frontal parietal network that has been theorized at the global workspace, for example, in the global workspace theory. So in the case of mind blanking, we seem to really have evidence that information is very much reduced at the level of sensory areas, and then that's makes it perhaps escape completely the global workspace, which could explain why they are not reporting anything. These are the sources of this thing, so you can see that we don't have at all the same global ignition in the case of mind blanking compared to on and mind wandering. So one of the issues with trying to track mind wandering and mind blanking is to know, since it's a private experience, to know if people are mind wandering or mind blanking, we have to interrupt them and ask them, which is pretty annoying because you're killing the very process you're interested in. So usually what we do is we have this random interaction and we ask people and they will report, I was mind wandering, I was mind blanking, etc. What we tried to do was to understand what was going on in between probes. And to do that, we train a machine learning algorithm. So taking these EEG features and trying to reconstruct what was going on. So this is an example of this reconstruction. So the color dots are the actual responses, the actual reports of the participants. And the others is what we reconstruct. So to make sure that our algorithm is working, we did two things. First, we did the classical training testing. We checked that the classifier is able to recognize trials that were labeled mind wandering, mind blanking by the participants themselves, and we're above chance. It's modest. I mean, the chance level is a third, and we're at 47%. So it's significant, but still way far from perfect. What was interesting is for the trials that were not labeled by the participants themselves, because they are too far from a probe, we looked at the behavior based on the label we reconstruct. And when we do this, we find the same pattern than the trials on which we have reports. So when we relabel a trial, mind wandering, mind blanking, we saw that they slow down in mind blanking, they speed up in mind wandering. So something very similar to the things on which they actually reported, which give us hope that this is going in a positive direction. Why we're interested in this? Because once you can move beyond probe, you can do a lot of interesting things about the dynamics of these states. You can compute how long they last, what is the probability to distribute the transition between these states. For example, a frequent question I have about mind blanking is, how long is a mind blank? And we don't know. It's very difficult to ask people how long they mind blank. But with this analysis, we have the impression that mind blank were short as mind wandering. Obviously, this is very, very far from an actual measurement of the duration of a mind blank event, but maybe it's going in that direction. So we think that this work has also potential interesting contribution to these debates about theories and mechanisms of consciousness. What we see is that mind wandering seems to impact more later stage of perceptual processing. And we have results that are very, very close to the global playgrounds proposal from Claire Sergent, while mind blanking seems to occur much earlier. I will not pretend that we're solving everything. I mean, when you look at the pattern of result we have, it's actually compatible with both the global neural workspace and the integrated information theory. It's very difficult, as if you have followed this space, to actually disentangle the two. I mean, there have been millions thrown to these attempts, and yet very often these theories say the same thing, but at least we are compatible with what they say, so it's a nice thing to have. Now, these sleep intrusions, so I kind of presented it as something occurring in a task. Usually this task lasts for 1 hour. What we try to do is to see what is the regulation of these events across longer time windows, for example, an entire day. And this comes from the sleep literature. I mean, we know that sleep is related by time. The more you spend time awake, the likelier you will be to fall asleep. But there is also a circadian regulation of sleep. There are moments of the day or the nights where you are more likely to be asleep or awake. And sleep is also use dependent. If you have done a lot of mental activity, you will also feel a bit tired and you will sleep deeper. So we wanted to see if these different regulatory mechanisms are also present in wakefulness. So we started with the two first one. It was a collaboration with Claire Anderson, who was at the time at Monash University. So Claire Anderson studies sleep deprivation and the consequences of sleep deprivation. So she was doing a very long protocol, 26-hour sleep deprivation. And I kindly asked if I could put a salt into it. So we had very, very merry participants doing 26 hours of sleep deprivation. Every 2 hours, they had the cognitive battery, including a salt with mind-wandering probes. When you look at how sleepy they are, so 9 is actually very sleepy. I keep forgetting to change this axis. The increase in sleepiness over time is not linear. So it's not like they gradually became more and more sleepy. It's actually, you have a first increase, then they get better. It corresponds to their evening. And then this is a night of sleep deprivation where they get gradually worse. You see it behind the square, but actually toward the morning, they get better again. So this is just the reflection of two influences. homeostatic influence. So the more you spend time awake, the more you're sleepy. But there is also this circadian sinusoidal influence that will help you being a bit better at the correct time window. So we modeled this homeostatic and circadian influence based on these vigilance ratings, and we try to look at their respective influence on behavioral aspects, mental states. And what we saw is that both homeostatic and circadian influence impact pretty much every variable you can see here. The exception, interestingly, is mind wandering, who in our data was only impacted by the circadian rhythm and not the homeostatic regulation. When you think of the parallel between mind wandering and that dreaming is also circadianly regulated, maybe there is something to it. We have a marker of slow waves. We wanted obviously to know how slow waves were impacted by both this homeostatic and circadian influence. And you can see that they are impacted. And again, these are slow waves recorded during wakefulness. What was interesting is we have different maps for this different type of influence. For the homeostatic process, we seem to have mostly an impact on frontal slow waves. This is quite interesting because this has been actually reported in the sleep literature, and we know that the prefrontal cortex is very sensitive to homeostatic pressure. So when you spend a long time awake, that's your executive control that's going to be damaged, not really your ability to perceive the environment. Circadian influence was different because it also impacts the frontal electrode, but also the occipital ones. So for the occipital electrode, when they show slow wave, this seems to be more associated with a position in the circadian rhythm cycle, than how much people have been spent awake. So as if there were different regulatory mechanisms depending on the location of these neurons. These slow waves were able to predict behavior again, and here mostly mind blanking on top of this homeostatic circadian influence. So they are interesting to track because they give you more personalized information about what people are going to do, what people are going to report. This is actually, we think it's related to the fact that sleep and attention share neuromodulatory pathways. And so what we believe is happening is if you take a system like dopaminergic and noradrenergic modulation, you can have large swings of this neuromodulatory pathway, and that can trigger big shifts, global shifts in neural dynamics from wake to sleep. But you can have also smaller fluctuation, either during wake or during sleep, that will determine if you're awake, whether you're paying attention or not to your environment, if you're asleep, whether you're sleeping well or not. To test that, I collaborated with Mark Belgrove, where we did a pharmacological trial, where we gave to healthy participants undergoing a sustained attention task, either noradrenergic and dopaminergic promoting molecules, so methylphenidate and aminoxetine, or a molecule promoting serotonin. And I will go briefly on that, but when you amplify noradrenergic and dopaminergic concentration, you have less slow waves in these individuals. And when you give serotonin, you have an increase in slow wave. So with neuromodulation, we can tilt the balance one way or another. We're currently replicating this on data sets that we borrowed from the lab of Yuval Nir. In that case, they focus on noradrenergic activity, and they use bidirectional pharmacological intervention. So using clonidine to decrease noradrenergic modulation, or reboxetine to enhance it. And what you can see from this side is when you do clonidine, so you push noradrenaline down, you push slow waves up, and the reverse when you give reboxetine, which could be perhaps one way to avoid having too much of these slow waves. So I talk about the time, the homeostatic circadian influence, but there is also a use-dependent aspect of sleep that we wanted to confirm with this wake intrusions. So what we did here was to have participants do very long tasks. So it was an entire day, 5 sessions throughout the day. And we had two groups of individuals. One group that was doing an easy task, it was a one back, and a group doing a hard task, which was a three back. So you have one group that will do a one back the whole day, one group doing the three back the whole day. You can imagine that at the end of the day, they are not in the same state. So each session was divided into blocks, and each block was made this way. You had the end back, and then at the end, there were economic choices to reveal fatigue. Because when you talk about money, it's when people reveal their true self. It's one of the best ways to know if people are tired. What happens with this kind of economic choice is when you're tired, you will go for the low costs, but also low risk option. So you want the money here and now rather than more later. So the task was made so that task performance was the same in the two groups. Fatigue rating was also the same, so they didn't feel more tired subjectively. But when you look at their economic choices, when they are doing a hard task, you can see that throughout the day, they become more and more, in a way, impulsive. What we did was to look at, again, our slow waves. So they are awake, they are doing a task, they're doing an end back actually, so they have their eyes open, they don't show any signs of falling asleep. But if you look at the normalized slow wave density, well, the people engaged in the hard task had a higher rate of slow wave increase, as if their brain was getting more tired and they have a buildup of sleepiness. And this is something that's more centrally distributed. There is an overlap actually with decision-related markers of fatigue. So we think that again, this wave has some impact on the way that people make decisions, which is something that we saw here. So this is an interaction between session and difficulty. So the fatigue factor, and so it's correlated with that. But they also correlate with choice behavior once you have removed this fatigue factor. So if you look at the way people behave, once you have removed the influence of fatigue, this low wave is still informative about whether they are going to make an impulsive choice or not. So we think they are mechanistically related. We actually think that this work can be linked to results obtained in fMRI, where they saw with the same kind of paradigm and hypoactivation of the frontal gyrus. So this hypoactivation will actually track very well with an increase in slow waves because they're usually associated with less activation. And a recent study from our collaborators at the Paris Brain Institute also showed that doing the same task, you have metabolic changes and you have an increase, a local increase in glutamate when you're engaged in the hard task. So what we think is we could have actually some sort of very holistic model where when you're doing something hard, you use your neurons more. So it's very, very basic thing, but these neurons, they burn energy, they produce metabolic waste. This metabolic waste could themselves trigger the occurrence of slow waves because we know that slow waves are involved in not necessarily metabolic clearance, but at least when there's metabolic waste, you have more of this slow wave. So it could be just a local regulatory mechanisms where if you use certain areas more than others, they will produce more waste and you have to get rid of that waste, or that waste is harmful. So maybe you switch to a slow wave-like activity to avoid that. So we have a lot of links between these wake slow waves and the regulation of sleep. So it really, comforts this notion of a continuum. Now, very, very quickly, because I'm time, but I wanted to show how this work can apply to clinical populations. And this is very much because now I'm in the Paris Brain Institute, and the Paris Brain Institute is in a big hospital. It's actually one of the birthplaces of neurology because where Charcot and other very famous neurologists did work their whole career. So being there give me access to a lot of clinical data, and sleep being related to pretty much everything. It's a fun playground. So this will be just very, very briefly, but a few examples of how you can apply this to a clinical population. So the first work is actually done when I was still in Monash with Mark Belgrove, and I recently graduated in PhD student, Elaine Pindle. We looked at ADHD. Why? Because ADHD frequently reports sleep disorders. The occurrence of sleep disorders in the ADHD population is 50%, so it's very high. So what we did was to look whether slow waves, whether ADHD have more slow waves than control, and they do. And these slow waves partially explain actually the attentional disorders. So patients with more slow waves will have more attentional deficits. What we believe, but we still have to test it, is that the sleep disorders could actually have an impact during the day. If you're not sleeping well, you're not doing a lot of slow waves during the night. So you will have the tendency to catch up during the day because you remember there's this homeostatic regulation. So more slow waves during the day means less attentional capabilities. We're also interested in neurodegenerative disease for the same reason. I mean, neurodegenerative disease like Alzheimer's are usually associated with sleep disorders, and there is a lot of evidence for bidirectional relationship between the neurodegeneration and the sleep disorders. So we took a big data set of hundreds of patients that were followed for 5 years, and we looked at whether slow waves were predictive of two things, either the amyloid deposits or the neurodegenerative, the neurodegeneration, two things that were estimated with a PET scan. And what we see is that actually people with either amyloid deposit or neurodegeneration or both have less slow waves but of bigger amplitudes, which was not necessarily what we were expecting, but kind of makes sense. When you're aging, your brain is less able to make slow waves, and that's what sleep deteriorates, unfortunately. So it makes sense that during wakefulness, they're also able to make less slow waves, but these slow waves are of higher amplitude, especially in this A plus N plus, so the one that have both amyloid deposit and neurodegeneration. So it seemed that there are fewer slow waves, but larger, and that could be perhaps associated with some cognitive deficit. At this stage, we're mostly interested in the biomarker level. It's not just about necessarily sleep disorders. These slow waves can also occur in the context of brain lesions. Actually, the first papers on delta waves were brain tumors papers, because before we had MRI, we used EEG to locate brain tumors, and clinicians noticed that post-mortem, obviously, that there was some sort of localization about slow wave they saw in the wave EEG and the localization of the brain tumor. And this is occurring in any type of brain lesion, so stroke, inflammation, tumor, etc. With Marcello Massimini, we wrote a review about that. These brain lesions, they have impact beyond the lesion area because they are connected to other brain regions that will be impacted, a phenomenon called diaskisis. So you can have regions that are not necessarily affected by the brain lesion, but connected to the brain lesion that will change neural dynamics and express the way. So what we try to do with Mark Debrov is to see if this is a kind of inversion we could even access with non-invasive recordings. So we looked at the EEG of stroke patients, and stroke patients have more slow waves during wakefulness than equally old but healthy patients. We couldn't see, however, some sort of localization effect. So if the stroke was on the left, it's not like we saw more slow waves on the left, or if the stroke was on the right, more slow waves on the right. And this could be due to the fact that at the scalp level, you have volume conduction. It's very hard to pinpoint the source of the activity. But again, it could be an interesting biomarker, and it correlates somewhat to some of the behavioral deficits in these patients. We believe slow wave intrusion come in two kinds. The normal kind, so the one that I presented so far, when you get a bit sleepy, your brain will do a bit more slow waves, and that will impact your attention, your stream of consciousness. In some cases, it can go beyond these kind of normal fluctuations. And we think it's a different kind of sleep intrusion. It's the kind of things you will see, for example, in Parkinsonian patients. They have illusion or hallucination that get worse and worse with the progression of the disease. And they have also a lot of sleep disruption, especially REM sleep disruption. So with the collaboration of the lab of Olaf Blanke, we look again at EEG of these patients, and what we saw is that overall Parkinsonian patients have more slow wave than healthy controls. But the one that experience hallucination have even more slow waves during the day than Parkinsonian patient without hallucination. So it's a biomarker of the occurrence of this hallucination. These slow waves are different, they're a bit faster, so they look more like the kind of thing you will see in REM sleep. So we think it's also a different kind of slow wave intrusion and more of a REM intrusion. And this REM intrusion will trigger a dream intrusion as well. So they're dreaming in their real life, and that's why they hallucinate. So it's not mind wandering, it's something quite different. Which is also something we see in narcolepsy. So if you watch TV and Severance, you have this wonderful scene that is, I think, a very, very nice depiction of hallucination in narcolepsy. You're sitting at your desk and suddenly something strange happens. We collected data on narcoleptic patients in the lab. So same thoughts as before, just that this time we gave the option to report I'm hallucinating or I'm having an illusion. And interestingly, Parkinsonian patients report that quite a lot in these thoughts and not the healthy individuals. You could think it's a report bias. I mean, they're narcoleptic, they know they hallucinate. But when we look at what happens before they report this hallucination, we see an increase in theta waves compared to all other states, including mind wandering. So again, something that looks like sleep, but this time non-REM sleep, and that could explain why maybe they're suddenly hallucinating. And that was it. So just as a very, very brief recap, I mean, your stream of consciousness is shaped by your brain activity. I mean, there is nothing new here, but the sleep-like wave seems to be a very important actor in that. They are modulated by the exact same regulatory pathway that will regulate sleep, which means that we can act on that. And they can be either a biomarker or a mechanism of a lot of neurological or psychiatric disease. But beyond sleep influence, I think we can now perhaps generalize these slow waves to something that is not just sleep related. I show you with a stroke result. These slow waves can also occur because of an accident. And in neurology, at least in the hospital where they work, they call that cortical suffering. So each time your cortex is suffering, its reaction will be to make these slow waves. So they are present in case of inflammation, brain lesion, metabolic stress, etc., etc. So they seem to be something that, again, in the normal case, occurs because of sleep pressure, but in some pathology cases can occur because of something else. So going back to the initial picture, if you look at sleep and wakefulness, not as separate states, but as a continuum, you can explain a lot of things by looking at both the global and local regulation of brain activity. And there is potentially a lot in common between what happens during the day and what happens during the night, which is something that has not escaped people before us. There's this wonderful quote from Victor Hugo. In French, mind wandering will be translated as rêverie, which is very close to rêve semantically. So I think, yeah, we're just rediscovering something very, very banal almost, that this modulation of consciousness during the day have everything in terms of relationship with what happens during our night. So thanks a lot. That's the dream team. So the basic science part and the clinical part, and all the people can get money, including the social environment. Thank you. Sorry that was a bit long, but I'm very happy to listen to you.
Thank you very much, Thomas, for delivering an excellent talk despite our monitor mind wandering, being on task 50% of the time. So let's open the questions to students in early career. As always, do you guys have any questions to get started? Yes.
I was just curious about I do a lot of research in circadian rhythms, especially in epilepsy. So not really related, but I was curious about your research into circadian rhythms and whether or not you've looked at morningness versus eveningness.
No, so I'm not really a circadian researcher, and I think that's what we do in terms of circadian regulation is clearly words away from what we should do if we wanted to look at that precisely. I mean, we should do like full desynchronization protocol and so on. That's a bit complicated. to do. So what we have is, some influence of the circadian rhythm. And we have this through this sleep deprivation procedure. When we look actually at morning versus evening differences or morning indecisiveness type, we never found anything. And I think the reason why is we don't control things enough. We have these very broad categories. We do the tests at fixed hour, whatever time people woke up or went to bed. So I think we're just lacking like the proper protocol to explore that. And yeah, it's a bit difficult to do it when you're not the proper lab setting.
Time for a couple more questions. Yeah.
Thank you. That was a lovely talk. I'm interested in how you think this applies or can apply. Have you done any studies in insomnia? So, you know, when I know Leon Lack did a study where they woke people with insomnia after 1 or 2 minutes of non-REM sleep and said, just before you heard the tone that woke you up, were you awake or asleep? And they all said, oh, we were awake. It's like, can this go the other way? That like maybe that's alpha, you know, even though their surface EEG is wakefulness. What do you think's sort of happening?
No, so I presented here, let's say half of my research was on wake. There is another half on sleep, and a lot of it is on actually insomnia and hypersomnia. And what we see is, the mirror image in insomnia. People with insomnia have intrusion of wakefulness in their sleep. More complexity, all the things that's reduced in mind ordering and mind blanking, you see it up in people with insomnia. There's a work of Francesca Ciclari that did these experiments, waking people and asking them, and when they say, oh, I was actually not sleeping, what she sees is wake-like activity buried within sleep. What we think is really like the mirror of each other, and everything that I said here can be applied in opposites to insomnia. That's kind of the characterization of insomnia. Now, can you do something about it? That would be wonderful, especially for people with insomnia. We haven't published data with Claire Anderson, where we did this approach of trying to boost slow waves in sleep with a tone synchronized to the slow waves. And what we saw is that boosting slow wave in sleep has limited advantage in terms of behavior, but it reduces slow waves in wakefulness. So we really have this homeostatic regulation that perhaps if you do that long enough, that can help. The other question is how you reduce fast activity during sleep, and that's much harder. I don't think we have an answer for that yet.
Yeah, Leon, who was doing this study with Jeremy Mercer, they thought they'd try and just retrain people that actually know you were asleep. And so they did a second study and said, well, actually, before you heard the tone, we could see that you were asleep. And these insomniacs would just argue with them through the intercom. I was not, your computer must be wrong. I was definitely awake. That's not going to work.
Discussion. I think one thing we started to do, and it's difficult, but I hope that someday we'll be able to have a kind of a good solution for that, is to sonify the EEG, so that people can listen to their brain, because we don't have feedback on our brain activity. I don't know how much slow wave I have right now. I don't know how much alpha. I can infer that with feelings, but I don't have that information. If you sonify EEG, you could perhaps give feedback to people about their brain states. And perhaps if it's not someone telling you you were asleep, but you inferring it based on what you listen, perhaps that would be more acceptable. And perhaps that could help people recognizing, okay, at least I'm going in the right direction. I should not stress about being tired tomorrow, etc., etc. That's would be lovely. We have a consortium actually in Europe called the Lullabites. It's funded by the Marie Curie program, and it's a set of 10 labs working on the relationship between sleep and music. So there are both sleep labs, music labs, and we're trying to find actually practical solutions to problems like insomnia.
Okay, we are at time now. We have a couple of questions still. Perhaps we can go into a little coffee break here, and you can ask Thomas all the questions that you have. Again, let me thank you, Thomas, for the beautiful talk, and again,